Dear friends, in the previous video we discussed about mechanism of auxin action. Now to continue our interaction we are going to explain what is Giberllin signaling. The concept explains how the Giberllin synthesizing genes gets regulated. To show you how the whole genetic pathway is driven, let me explain some key components used in the mechanism. The first and foremost point to understand here is known as ligand. Now what is the ligand? It is Giberllin molecule which acts as a signaling molecule. Now what is a receptor? Receptor is a special type of protein used in the whole pathway. The receptor has special type of ability, it binds with the ligand. The receptor used in this mechanism is known as GID1, means Giberllin incentive dwarfin protein found in the rice. It is a soluble receptor protein. However, in Arabidopsis, the receptor has three important orthologs, namely GID1A, GID1B, accordingly GID1C. It was all about the receptor. The receptor has special type of active sites to bind with the Giberllin during the gene expression. Now to proceed, let me tell you very important key component used in the mechanism. This is known as transcriptional factor. As the word indicates, it is used in the process of transcription. Here the transcription factor used in the whole pathway is known as PIFS, means phytochrome interactive factor. Now coming to the next very important protein, this is called a repressor. As the word indicates, it suppresses the activity of transcriptional protein and acts as an inhibitor protein. The repressor plays significant role in the whole pathway. It fuses with the transcription factor and stops the process of transcription of Giberllin synthesizing genes. The repressors used in the whole process are known as delaproteins. These delaproteins have two important types used in different species: SLR1, cylinder rice 1 protein as in rice, GAI and RGA. These are two different types of delaproteins found in Arabidopsis. Now one more important family of proteins used in the pathway, these are called F-box proteins. These are target proteins. They targeting protein degradation in cell. They add ubiquitin to the delaprotein by the process of polyubiquitination. Now last point of terminology, this is known as proteasome 26S type. It is a protein complex. It leads to degradation of misfolded, unwanted or damaged proteins present inside cell. This proteasome has special type of structure. It is made up of core part and two caps, lower and upper. Core part has two alpha rings outer side and two beta rings inner side. When the delaprotein, it is tagged with the help of ubiquinone by polyubiquitination in presence of ubiquinone ligase. This delaprotein enters into the proteasome and it leads to lysis of delaprotein. It was all about terminology. Now coming to the mechanism, what happens actually? Mechanism involves two important pathways. In pathway number first, when the ligand is totally absent, as shown in the fig, suppose this is DNA and it has Giberllin synthesizing genes. Now later on, these genes get expressed only in presence of transcriptional factor known as PIFS. But in case number 1, when ligand is present, the delaprotein fuses with transcriptional protein and stops the whole expression of Giberllin synthesizing protein, means transcription stops and the whole operon stays, what we call it as off. Right? Now proceeding to the next very important pathway, when the Giberllin is present. This pathway is opposite to the first pathway. Here the ligand or Giberllin is present. Then what happens? Suppose this is DNA. It has Giberllin synthesizing genes. And here this is known as GID1, means receptor. and ligand fuses with the receptor, and again delaprotein here fuses to form GID1-GA delaprotein complex. Then one more group of protein combine here. These are called abox protein. They lead polyubiquitination tagging of delaprotein. Then this delaprotein enters into a proteasome as discussed earlier. This proteasome leads to lysis of delaprotein. It forms peptides. Now the transcriptional factor here present is totally free. It is in active mode. It leads transcription of Giberllin synthesizing genes which form proteins, and proteins form well-defined Giberllin. After Giberllin formation, physiological activities takes place. Suppose some physiological activities of Giberllin includes: it helps in plant body seed germination. It performs the function of leaf expansion. We can also add it helps in stem elongation, seed development also. These are some important functions of Giberllin. I hope you got the concept beautifully. Thanks. Have a nice day.